this afternoon. Smithfield Foods is about a $14 billion company here in the U.S., owned by WH Group in Hong Kong now. And we do produce a lot of packaged meats products as well as fresh pork products. The larger portion of our business is in packaged meats, processed meats, and have a lot of retail brands, but also sell to the food service industry and to restaurant chains and so forth. And so one of our principal focuses for processed meat microbiological control is food safety. And of course, when you're talking about ready-to-eat meats, that certainly involves and heavily involves, if not principally, Listeria monocytogenes. So as you know, this is a gram-positive bacterial pathogen. It's got a very, has a high mortality rate once a person becomes infected. We have to be concerned about it in the meat industry for its, because it's essentially selected for in a cold, wet processing environment. So once you've produced a cooked or a fermented or any kind of a ready-to-eat meat product, getting it into the final package is at the point where you always have to be concerned with preventing contamination of this organism. But there's two approaches. We have sort of a dual approach. That is controlling it in the environment, monitoring it in the environment. And the other is formulating products or processing them in some way to mitigate the organism if it were in fact present. And to do that with formulated inhibitors and combinations with sodium chloride, we're considering the growth limits of the organism with respect to pH and water activity primarily. And so for microbiologists, we do prefer to use water activity as our measurement of water. It's essentially the biologically available water. And we consider the pH and, of course, the ingredients. So in my role, I work with our product developers, our R&D groups, to make sure that they're formulating products to control growth of the organism, to restrict the growth, and thereby reduce risk to the consumer by keeping any incidental contamination in the lag phase during the refrigerated storage. In other words, we never want it to come out of lag phase. And we do that with salt and organic salts, such as sodium and potassium lactate, sodium diacetate. And of course, sodium nitrite is used heavily in processed meat products to control Clostridium botulinum and to impart that characteristic cured meat property. These are the organic salts that are most commonly used, and probably around 2000, 2003, the U.S. meat industry went almost full bore into the use of these types of ingredients to control the growth of Listeria monocytogenes. And of course, as you see, two of these organic salts have sodium as a cation. So they do contribute, as was mentioned previously today, to the sodium in the meat product. But potassium lactate also came into use pretty heavily, and as long as you could get around the metallic off flavor, that would be certainly within the realm of usage for product developers. These come in commercially available ingredients produced by a number of different ingredient companies here in the U.S. and worldwide. And by and large, the majority of ready-to-eat meat products here in the U.S. are formulated with about a 56% to 4% sodium diacetate, or potassium lactate diacetate, and then sometimes a combination of sodium and potassium lactate. And more recently, potassium acetate has come on very strong to help the industry get away from the use of sodium. Potassium acetate is actually approved as sort of a flavoring, so it can be labeled as flavor in some cases. So it's actually a flavor enhancer in some food systems, and it is efficacious. So the mechanism of action is essentially twofold, I would say. Of course, there's water activity reduction, which helps control growth. But also, if these molecules go into the acid form, they can penetrate the bacterial cell membrane and increase and then dissociate once they're inside and disrupt the pH or the hydrogen ion balance between the extracellular fluid and the cytoplasm. The cell has to compensate by continuously pumping out these hydrogen ions and thereby expends its energy doing that rather than using its energy to grow as a population. Thereby it stays in lag phase. But this research by Cornell gave much better insight into other mechanisms, and they looked at combinations of lactate and acetate on transcription of Listeria monocytogenes genes and showed that when lactate and acetate are present, it shifts the balance of transcription to the fermentative metabolism and production of acetoin. It's almost like a feedback inhibition mechanism. So that's a great study if you want to find out more details. The industry has gone largely over to clean label. That's a really common request we get from marketing people, as well as reduced sodium or lower sodium. So clean label means typically the consumer doesn't want to see, or the customer in some cases doesn't want to see X, 8s, and ites. And so vinegar is one way we can get organic acid into the product with that perception that it's clean and natural and so forth. So my role in all this is to take the product developers and guide them through their process and then bring the product samples to our lab in Cincinnati and do a challenge study, essentially. So we maintain Listeria monocytogenes stock cultures, and we basically use those to inoculate in the product. We obtained a couple of the ones that we use in our standard challenge study inoculum from Cornell, and they were part of an ILSI project. They have an ILSI number. They were associated with outbreaks from ready-to-eat meat products. So we essentially take those strains and a few others in a plant environmental isolate. We inoculate products, we vacuum package or gas flush them, we simulate what would happen in the plant. We then store these and plate them out over time on selective augers and recover the inoculum to determine the growth. Okay, so the first case study is in a deli meat. And this is a product that was in the marketplace. We were treating it on the surface of the product with antimicrobials as a post-lethality treatment. Therefore, it didn't have any other formulated inhibitors other than a lactate and diacetate blend. But the customer, who is a very large retail customer who slices this and serves it to consumers, wanted to have their delis protected. They wanted their consumers protected when they brought the packaged deli meat home. So they asked the industry to provide even more enhanced growth control of Listeria monocytogenes. And to do that, we basically used a combination of what was already in it, the lactate and diacetate, but then we added lemon juice and vinegar product. So we're putting citric acid and acetic acid into that formulation. And essentially the challenge study revealed that the no inhibitor control grew quite well during storage, and the formulated inhibitor product suppressed growth of Listeria monocytogenes well into the shelf life and even farther than we needed. The other example is a retail quick serve restaurant customer who is purchasing what is, I think, qualifies as a low sodium ham. And they would receive it from us as a supplier in a modified atmosphere pack with slices, and their people in the stores would be able to open up and put it on sandwiches. So this product was being it did have formulated inhibitors, but it was also being treated with high pressure. So it had essentially the redundant system, if you will, the killing of any potential pathogen that could be there and the growth suppression over time. So we had approached them about perhaps removing the high pressure because it's a very expensive process and time consuming. And so we developed a few, the R&D group in our innovation center and pilot plant in Smithfield, Virginia, developed some prototypes, and we put together a challenge study with a no inhibitor control. We replicated our proprietary or our proposed ingredients. We did 2 replicate production runs of that with 0.6% dried vinegar. We used another commercial product ingredient that had 0.6% vinegar that was a product of fermentation, and then a buffered vinegar liquid ingredient. Each of these, we targeted this level of sodium chloride on a percent basis and sodium per serving per 100 grams. And the pH and water activities are listed there. And again, we did show suppression of growth of Listeria monocytogenes with all of these formulations. So it essentially shows that this product did not necessarily need high pressure processing, that growth control is probably sufficient to protect the consumer. Last slide is lactic acid bacteria. In that same study, we also, as a standard practice, we prepare uninoculated samples and monitor the spoilage flora. And this slide demonstrates that there is slight difference in some of these variables and a significant difference in some of the others. So there are some other factors at play microbiologically than pathogens. And that's all I have. Thank you.
Well, we heard of some good options to use. What about this dried vinegar? Any issues with spoilage microbes?
I think in one of those variables, we did 2 replicate variables of the same dried vinegar. And in one, we saw growth of lactic acid bacteria, and one we did not. So it's kind of hard to gauge until you get into industrial production, where you're then dealing with the actual microflora of the environment. But overall, I can say that when the industry switched to lactate diacetate, they did see an enhancement of shelf life. So one would think that should also come, for the vinegar products as well.
Okay. Thank you.